pharmacogenetics, immunogenetics, epigenetics approaches. With this brief introduction, I welcome Dr. Mainak and for floor is yours, sir. Thank you.
Thank you, Mustaq, and thank you, Gyaneshwar, for inviting me for this interesting meeting on pharmacogenomics. And I have two major reasons for cheering up because my mentor, Professor Rajiv Singh, was here as PhD student and then subsequently went on to be the Vice Chancellor. And the second reason is that my father's elder brother, who also happened to be an Professor Emeritus in Archaeology Department of BSU, and who went on to become the Director General of National Museum, who has written several books on Iron Age of India there. So therefore, there is a lot of interest, particularly more excited on this particular occasion in BSU and give this talk. But my talk is completely different than what we have been hearing since morning, and it's quite different from what Prof also gave. So therefore, I will try to skip on how to skip the slide. Up and down. Okay. Yeah. So good. So I'm trying to talk about the dilemma and epigenetics of neuropsychiatric diseases and the way to resolve it. So my lab basically works on genetics of complex disorders of CNS and mostly non-communicable diseases. And the disease that I work on, there is no biochemical phenotype and there is no symptomatic phenotype. So therefore, it's completely left to the judgment of the clinician who is doing the judgment there. And so therefore, whenever we talk of any such disease, it's heavily influenced by genetic diversity. It's also heavily influenced by phenotypic diversity, and it's also influenced by epigenetic diversity. And therefore, we really need to stratify all these aspects before we really get into the disease genetics or disease epigenetics. But I'm not going to talk about genetic stratification and genetic phenotypic stratification today. I'm mostly concentrating on the epigenetic stratification. And as we all know that epigenetic stratification basically requires, we know, and I'll take an example with schizophrenia as a case study. And we know that schizophrenia is heavily influenced by gene-environment interaction. And we also know that yes, children born in the winter months are more prone to develop schizophrenia. But the issue is that all children who are born in the winter months are going to develop schizophrenia. So that's what we are going to try to address it. So it is not true that every child who is born in the winter months are going to develop, and why? I try to address that particular issue. And so these are the molecular markers of epigenetics. One is DNA methylation, other one is microRNAs, and these are the mature markers of epigenetics. And the third is histone modifications. I will try to cover two aspects today on DNA methylation and microRNA observations. And when we started working on epigenetics or DNA methylation on schizophrenia, there were plenty of papers that have been existed in the various public domains. And therefore, we were very curious to know how much of these signatures that have been reported to be signatures of pathogenesis are the real signatures of pathogenesis. And therefore, we literally wrote a perspective article questioning that is it that those markers are real signatures of pathogenesis, or they are they influenced because of genetic heterogeneity, or they influenced because of the medication issues? Because most of the studies that have been performed till date, we published this particular paper, it was mostly on the patients who were on conventional treatment protocol for schizophrenia. So it means that they were already into medications there, and a good amount of study has been done on the postmortem samples there. So it means that the patient must have actually died because of the drug issues also. So therefore, we try to address two aspects, and then we went on to publish series of evidences on that why we say such things, which I'll also show you by evidences there. So therefore, we question that how much of these epigenetic observations because majority of the studies that were performed were based on case control studies. And does ethnogenetic background to play a role? And studies were, besides the studies were based on postmortem samples and do antipsychotic influence the methylation observed. So when and if you literally see, most of the studies were actually performed as the case control studies. And when we see the case control studies, as per this particular data, it would look like hypermethylation is basically a state of schizophrenia, is a state of schizophrenia pathogenesis. And similarly, hyperhomocysteine possibly might be an indicator of schizophrenia, as an indicator of schizophrenia. But is it really true? So I'll come back to this slide quite often. And therefore, what we did is we again took the public domain data, which was again on schizophrenia, and particularly from the American data sets. And what we did is we tried to compare, and that particular data set had both white American and also black Americans. And we tried to certify this, how much of these white Americans and black Americans within the control data are heavily influenced in the DMPs. And you can see that here, this is about the control data sets. You can see that yes, there is a huge amount of differences between the control data sets between the white Americans and black Americans. And similarly, when you compare the schizophrenia data set between the white Americans and black Americans for the same, you can see that there is a huge amount of differences between the even in the schizophrenia data set. And not only that, the sites which are reported to be hypermethylated in white Americans, it has been reported to be hypermethylated in the black Americans. So similarly, there are several series of discrepancies between the data sets itself. So these are all the data sets which have been reported with respect to pathogenesis markers for schizophrenia. Now we dissect the same data set and we say that no, there has to be something else behind it. So therefore, we know that yes, all methylation data are actually likely to be influenced because of the genetic backgrounds, and therefore that really takes us to investigate what are the drivers for methylation there. And therefore, we know that the drivers of methylation are the endogenous mechanisms, are the denomethylation mechanisms, which endogenously control the denomethylation, demethylation processes, and then these are the exogenous mechanisms which requires the cofactors like the vitamins to really influence the methylation pattern. And for this one, the marker is the homocysteine. For this one, the marker is the global methylation pattern. So we checked for so then what we did is we tried to first see whether the genetics of methylation or DNMTs are really integral in the disease causation. Yes, we found in the case control studies, we found that the DNMTs themselves are genetically associated with the schizophrenia state. And the variants that were associated were actually indicative of the increased expression pattern, which means that the hypermethylation state possibly might be indicative of schizophrenia itself. And then, and therefore, I would say that yes, this particular indication would be possibly the indicator of the genetic states. And then when we try to compare, what does it really imply in the tissue-specific expression? And you can see that yes, in because mostly in the DNA, people will say that how does your observation really imply in the various body tissues? And you can see that the yellow one, which is actually been shown in the brain cerebellar regions, which is heavily expressing this particular DNMT1 expression there. And therefore, when you see that DNMT, what is its implication with respect to cerebral hemispheres, you can see that these are the regions which are heavily influenced, which are downregulated in the cerebral hemispheres, and which will further imply that the overexpression of DNMTs would result in increased DNMT expression, which will result in increased denomethylation, which will result in decreased gene expression there, the other gene expressions there. So therefore, the influence of gene expression patterns or neuronal gene expression patterns is possibly influenced because of the increased denomethylation patterns, which is influencing other genes which are mostly of neuronal lineage. And therefore, what it really implies from the global perspectives, we say that yes, the variants that we found to be associated are mostly is basically more reflective of the East Asian. So East Asian and South Asians actually have more or less the same allele frequencies in compared to the Africans and American populations. And similarly, when I do a phylogenetic observation, I see that yes, the Indians, basically where the Kerala population resides, the Telugu Indians and Sri Lankan Tamil, they are actually very close to the East Asian population. So similarly, when I try to look into the exogenous mechanism of DNA methylation, which is mostly on the methionine cycles, here again we see that yes, methionine cycle genes are again strongly associated with the schizophrenia cases, and therefore the increased homocysteine levels might be indicative of increased expression of this particular methionine cycle genes there. So therefore, and then comes is that are there such studies that has been reported with respect to increased methylation and increased homocysteine levels? Yes, there is a huge amount of meta-analysis studies that has reported that increased that hyperhomocysteinemia is a state of schizophrenia pathology there. But now comes the twist. The twist comes is that the antipsychotic drugs which are actually being used for treating patients, they themselves actually deplete the folate levels there. So depletion of folate levels will actually somehow increase the homocysteine levels, and that is where you tend to see that yes, this is a series of papers which show that these antipsychotic drugs do imply the folate levels, and so is true for many other drugs that we have been working on right now, which really deplete the folate levels. So therefore, hyperhomocysteinemia, whether it's a real state of the disease pathogenesis or they are really influenced because of drugs. So therefore, what we say is that it's very, very important that one has to really work out these DNFT variants or the methyl maintenance genes, which actually define the threshold of a particular individual living in the disadvantageous background are more prone to develop schizophrenia. So therefore, what I was initially saying is that not every individual who is actually born in the winter months are likely to develop schizophrenia. The definition of threshold is defined by its genetic counterparts. So now coming to the second twist is this is all about the genetics of the epigenome, and I did write in the editorial that genetics of the epigenome really is very integral to many complex diseases, not only for schizophrenia but many other diseases. And now comes the twist is that we then went on to work out how much of these observations are really influenced by the antipsychotic drugs. And so then we try to do series of such experiments using under a clinical scenario. It's extremely difficult to really find out the effect of each individual drugs there. So therefore, we have to try it out doing in vitro cell culture systems there. We use both liver cells and also the neuronal cells, and then try to see whether these antipsychotic drugs do influence the global methylation, gene expression patterns of these genes which are responsible for methylation maintenance in the microRNA profiling and also extra modifications there. And also try to look into gene expression patterns of epigenetic genes and pharmacogenetic genes, and then try to validate the observation that we get in the clinical scenario there. So therefore, these are the things that we did for global methylation, the genes that are responsible to maintain the global methylation, then the protein expressions, and of course, then we also did into the bisequence sequencing for following genes. We also looked into the pharmacogenetic genes, gene expression patterns. Then we also tried to look into the microRNAs that target these DNMTs and the microRNAs that target this drug metabolizing genes. And then we also tried to do the genome-wide mRNA expression analysis and also genome-wide genome expression analysis using H62 analysis. But it was not easy because when we started working on this in vitro cells, so we literally stumbled into another big mess because we were working on a series of experiments using this IMR32 cells, and ideally what the various publishers or various committees suggest that we should really do the DNA profiling. And fortunately, I got the same cell lines from two different laboratories, and we found that the DNA fingerprint profiles of the same cell line procured from two different laboratories are distinctly different from each other. This is the HCC, so this is the reference cell line and profile, and this is what the people claim that they have procured from HCC, and this is what the profile that the people claim that they have got it from a Rican brain bank there. The DNA profile is significantly different. That is what the routine journal says there. But what we did is we went on to investigate further. Is that the housekeeping gene expression profile is also the same or not? Housekeeping gene expression profile for many of these housekeeping genes that even when you try to look in the DNA methylation pattern, you can see that the DNA methylation for ATCC is close to 3% and for the EKIN is close to 1%. So, yes, yes. So then I quickly go. Then we went on to see that whether these antipsychotic drugs do influence methylation. Yes, they do influence methylation, which is increased DNA methylation. They also influence the increased gene expression. They also influence the increased DNA methylation, and they also decrease the microRNA mid-29B, which is the target for the DNMT1 there. So this is what in summary. And then we went on to this I'm skipping. We went on to prove under in vitro condition, under validation of the clinical setting. So we had a set of patients who were considered responders and non-responders, and we actually almost see exactly the same observation that we get in the under the in vitro observation, that there is increased DNA methylation. We tend to see there is increased DNMT1 expression. We also tend to see there is an increased and there is a decreased DNMT29B microRNA 29B expression there. So which means that decreased expression of this MYF29B might possibly imply this particular thing, but we still don't know what happens in why in the non-responder there is a further depletion there. So this can happen in the clinical scenario there. So therefore we say that this increased methylation state might not be the real indication of the pathogenesis. They might be the indicators of drug response. Because whenever we have a pool of samples, we will have largely the responder state individuals. So therefore, that is one issue. Then we went on to look in the pharmacogenetics. Yeah, yes, sir. I just finished. So same observation we get in the pharmacogenetic genes, and same we try to validate in the clinical settings. And then we went on to check for the microRNA for the global methylations. And what we do is when we look into the microRNA for the 850 methylomarin, what we see is that all these studies that have reported the various DMPs to be implicated in pathogenesis markers, the same observation we try to see in the drug response there. So the drug response markers actually really are overlapping with the pathogenesis markers. So therefore, we have to really take a word of caution from these observations. Same is true for the microRNA expression. So when we looked into the microRNA expression, these were the observations that were the microRNA expression studies that were reported till date, and we said that these microRNA are likely to be influenced because of drugs. And then we tried to work out with the microRNA expression patterns, and what we see is that here there is an overlapping expression of microRNA observation that we get with the antipsychotic drug, and the direction of microRNA expression is more or less the same. The yellow ones which you see here are the microRNA expression where the direction is the same. So if the direction of microRNA expression is the same, it means that they are not the hallmark of the pathogenesis. They are the hallmark, possibly they are influenced because of medication issues. They could be the hallmark of the drug response itself. So these are the real conflicts that we try to dissect it out in the published data, and what we try to validate. And this is also published in Frontiers and Molecular Science. With this, I will try to skip, and one could really say that is it really true how much is known about the drugs? Lot is clearly not known about the drugs. 91% of the drugs that is the PSO are not been tested for steroidal potential there. And therefore, even so, if you see that receptor defects and cleft lip, which we actually continuously work for genetic causes, they are actually influenced. They are actually mediated by cause of anti-epileptic drugs there. So we have another set of data which are not present here today on the anti-epileptic drugs. So thank you very much. I'm not saying all this.
Thank you for giving very illuminating talk on epigenetics of neuropsychiatric disorder. Due to lack of time, I think you can ask questions in the lunch hour because we have to restrict in time. Next speaker.
Thank you so much, sir. I request Professor Mustaf to come over and present Dr. Banarjee with the memento.
Thank you very much.
So we move to the third speaker of the session, which is the last